the lab experiments that happened in the Wuhan Institute of Virology. We know what happened with COVID-19. We know that the bat lady got money indirectly from Dr. Fauci. Dr. Fauci at the NIH gave a grant to Peter Daszak at EcoHealth Alliance, who subcontracted this gain-of-function experimentation at the Wuhan Institute of Virology, and Dr. Shi, the bat lady, conducted these experiments. Those experiments juiced up the virus, made it more lethal, more transmissible between human beings. It turns out that these same type of experiments have been conducted on the monkeypox virus at the Wuhan Institute of Virology. There was a study that was published in a journal called Virologica Sinica, and the authors of this article in this journal, the authors are the folks at the Wuhan Institute of Virology, including Dr. Shi's counterparts. This is what the title of the research article is: Efficient assembly of a large fragment of monkeypox virus genome as a qPCR template using dual selection-based transformation-associated recombination. Okay, so that's a lot. That's a big mouthful of scientific words. This, by the way, was published just a few months ago. And one of the key laboratories cited here, the group of scientists that wrote this were from the Wuhan Institute of Virology. So, efficient assembly of a large fragment of monkeypox virus genome as a qPCR template using dual selection-based transformation-associated recombination. So the only part of that, the only words here that we really need to understand are that the monkeypox, it was the monkeypox virus, and they were assembling the genome of the monkeypox virus, the DNA, and they were assembling a template of the DNA of the monkeypox virus using a particular procedure called transformation association recombination. Okay, TAR is how that's sometimes abbreviated. Transformation associated recombination. Okay, so really the only critical part to this study or this research paper is scroll on down to page 5. At the bottom of page 5, this sentence. It's about this TAR procedure that they use. It says, however, this DNA assembly tool applied in virological research could also raise potential security concerns, especially when the assembled product contains a full set of genetic material that can be recovered into a contagious pathogen. The DNA assembly tool applied in this research could raise potential security concerns. That's a pretty big deal here. A pretty big deal because we know, I mean, based on the title, the actual title of this study or the title of this article is efficient assembly of a large fragment of monkeypox virus genome as a qPCR template using dual selection-based transformation-associated recombination, that TAR procedure. And that TAR procedure can raise potential security concerns because it can make a pathogen contagious.